everyone, welcome to my channel. In this video, we are going to see cholesterol metabolism, which is my last video in metabolism of lipids. Before continuing with the video, please subscribe my channel. Cholesterol. Cholesterol is found exclusively in animals, hence it is often called as animal cholesterol. It is amphipathic in nature since it possesses both hydrophilic and hydrophobic regions in the structure. Functions of cholesterol. It is a structural component of cell membrane. It is precursor for the synthesis of all other steroids in the body. It is an essential ingredient in structure of lipoproteins. Fatty acids are transported to liver as cholesterol esters for oxidation. Let's see cholesterol biosynthesis. Almost all tissues participate in biosynthesis. Major contributors are liver 50%, intestine 15%, skin, adrenal cortex, reproductive tissue, etc. Enzymes involved in biosynthesis are found in cytosol. Acetate of acetyl-CoA provide all carbon atoms in cholesterol. Reducing equivalents are supplied by NADPH. NADPH is obtained from HMP shunt. Energy is supplied by ATP. 1 mole of cholesterol is obtained from 18 moles of acetyl-CoA, 36 moles of ATP and 16 moles of NADPH. Let's see cholesterol biosynthesis cycle or pathway. There are 5 stages of cholesterol biosynthesis. First is synthesis of HMG-CoA. Second is formation of mevalonic acid. Third is production of isoprenoid units. Fourth is synthesis of squalene. And fifth is conversion of squalene to cholesterol. So let's see the pathway properly. Glucose is converted to acetyl-CoA by glycolysis and by the action of PDH. Fatty acids is also converted to acetyl-CoA by fatty acid oxidation or beta oxidation. This acetyl-CoA is converted to HMG-CoA. This reaction we have seen it in ketone bodies video. Acetyl-CoA undergoes the activity of thiolase and HMG-CoA synthase to form HMG-CoA. HMG-CoA is also obtained from leucine. Now this HMG-CoA undergoes the activity of HMG-CoA reductase to form mevalonic acid. Mevalonic acid undergoes 4 reactions, including 3 of kinase and 1 of decarboxylase to form isopentenyl pyrophosphate. Isopentenyl pyrophosphate undergoes reaction isomerase and transferase to give farnesyl pyrophosphate. This farnesyl pyrophosphate can form squalene, dolicol, heme A or ubiquinone. Squalene is formed by squalene synthase. The squalene formed then forms lanosterol by activity of epoxidase, hydroxylase and cyclase. The lanosterol formed undergoes series of reactions, about 19 reactions, and the penultimate product is 7-dehydrocholesterol, which on reduction finally yields cholesterol. So this is cholesterol biosynthesis pathway. Let's see regulation of cholesterol synthesis. First is feedback control. Cholesterol itself controls its own synthesis by feedback mechanism. Increase in cellular concentration of cholesterol reduces the synthesis of the enzyme HMG-CoA reductase. This is achieved by decreasing the transcription of the gene responsible for production of the enzyme. Second is hormonal regulation. Glucagon and AMP inhibit synthesis of HMG-CoA as well as promotes CAMP-dependent phosphorylation, which inactivates HMG-CoA reductase. Insulin, on the other hand, promotes synthesis of HMG-CoA reductase as well as inhibits CAMP-dependent phosphorylation, thus activating HMG-CoA. Third is inhibition by drugs. The drugs compactin and lovastatin are fungal products. They are used to decrease the serum cholesterol level in patients with hypercholesterolemia. Compactin and lovastatin are competitive inhibitors of the enzyme HMG-CoA reductase. Then HMG-CoA reductase is inhibited by bile acids. Fasting also reduces the activity of this enzyme. Now let's see the degradation of cholesterol. The steroid nucleus of the cholesterol cannot be broken down in humans. Cholesterol is converted to bile acids, excreted in feces, serves as a precursor for the synthesis of steroid hormones, vitamin D, coprostanol and cholestanol. Coprostanol and cholestanol are fecal sterols besides cholesterol. So let's see first synthesis of bile acid. Here cholesterol undergoes the activity of 7-alpha hydroxylase to form 7-hydroxycholesterol. 7-hydroxycholesterol then undergoes several steps to either form cholic acid or chenodeoxycholic acid. Cholic acid can combine with glycine to form glycocholic acid or with taurine to form taurocholic acid. Whereas chenodeoxycholic acid can combine with taurine or glycine to form tauro or glyco-chenodeoxycholic acid. Glycocholic acid can be acted upon by intestinal bacteria to give deoxycholic acid. Tauro or glycochenodeoxycholic acid can be acted upon by intestinal bacteria to give lithocholic acid. Glycocholic acid, taurocholic acid, then tauro or glyco-chenodeoxycholic acid are all primary bile acids, whereas deoxycholic acid and lithocholic acid are secondary bile acids. Synthesis of steroid hormones. This is the second thing. Cholesterol is precursor for synthesis of all 5 classes of steroid hormones: glucocorticoids, mineralocorticoids, progestins, androgens, and estrogens. Third is synthesis of vitamin D. 7-dehydrocholesterol, an intermediate in the synthesis of cholesterol, is converted to cholecalciferol, that is vitamin D3, by UV rays in the skin. Now let's see the transport of cholesterol. Cholesterol is present in plasma lipoproteins as 70 to 75% as esterified form with long chain fatty acids and 25 to 30% as free cholesterol. HDL and the enzyme LCAT are responsible for transport and elimination of cholesterol from the body. HDL is high density lipoproteins and LCAT is enzyme lecithin cholesterol acyltransferase. It is a plasma enzyme synthesized by the liver. The cholesterol ester forms an integral part of HDL. In this manner, the cholesterol from the peripheral tissues is trapped in HDL by a reaction catalyzed by LCAT and then transported to liver for degradation and excretion. This mechanism is commonly known as reverse cholesterol transport. Now, plasma cholesterol level. It is of great biomedical importance. Let's see hypercholesterolemia. This is increase in plasma cholesterol more than 200 mg per dl. It is seen in many disorders like diabetes mellitus, increased cholesterol synthesis, hypothyroidism or myxedema, there is HDL receptors on liver cells decrease. Obstructive jaundice, due to the obstruction in excretion, cholesterol excretion is decreased. Nephrotic syndrome, there is increased plasma protein, lipoprotein fractions. Hypercholesterolemia is associated with atherosclerosis and coronary heart diseases. LDL is positively associated with atherosclerosis and hence is known as bad cholesterol, whereas HDL is negatively associated with atherosclerosis, or its high concentration counteracts the atherogenesis and hence is known as good cholesterol. High BP, stress, smoking, drinking soft water, coffee, lack of exercise, obesity elevates serum cholesterol. Control of hypercholesterolemia. 1 is consumption of PUFA. PUFA will help in transport of cholesterol by LCAT mechanism and its excretion from the body. Second is intake of plant sterols and fibers. They inhibit intestinal absorption of dietary cholesterol. Third is avoiding high carbohydrate diet. Fourth is adequate changes in lifestyle like quitting smoking and doing exercise. Fifth is moderate alcohol consumption in chronic cases. Red wine is particularly beneficial due to its antioxidants besides low alcohol content. And sixth is use of drugs such as lovastatin. Lovastatin inhibits HMG-CoA reductase and decreases cholesterol synthesis. Cholestyramine and cholestyramine bind with bile acids and decrease their intestinal absorption. Clofibrate increases the activity of lipoprotein lipase and reduces plasma cholesterol and triacylglycerols. Let's see some facts regarding cholesterol. Dietary cholesterol is safe to consume. We can state this fact because research findings repeatedly indicated that there is no significant relationship between the consumption of dietary cholesterol and serum cholesterol. Thus, cholesterol has become the biggest medical scam of all time. Cholesterol is a friend, not an enemy. Cholesterol is a vital and universal component of every cell membrane on earth. Further, cholesterol is a precursor for the synthesis of steroid hormones, bile acids, vitamin D, etc. So the focus is now on the dietary trans fat and sugars, which are considered as culprits in the causation of heart diseases. So this is all for cholesterol metabolism. Here we end the chapter metabolism of lipids. Other three videos posted for this are fatty acid oxidation, that is beta oxidation, fatty acid synthesis and ketone bodies. So I have covered 70% of the chapter in the 4 videos. Please go through the videos which I have uploaded earlier. The link has been provided in the description. Thank you so much. Please share this video.